about apoptosis and other forms of cell death. In your last lecture, we covered cell injury because of external stresses and injuries. This time, we will look at activation of endogenous death programs. Now, apoptosis and apoptosis are two alternative pronunciations. The English tell me that it's apoptosis, so I will try to use that pronunciation. But by and large, I might use these two terms interchangeably. So before I start, this talk is based on personal experience. Data which I'm presenting would be properly acknowledged and quoted at the end of the lecture, or if it's a slide, the source would be quoted below the image. Last time, we looked at necrosis, which is cell death in living tissue by external injurious agents. Today, we are going to look at apoptosis, which is cell death by activation of internal death program. Autolysis is changes in cells and tissue after removal of organ or death of organism. And counter to what you might imagine, the changes are very different in autolysis from both necrosis and apoptosis. So autolysis is mostly self-digestion because of release of lysosomal contents, and autolysis renders organs and tissues almost liquid or autolyzed or soft. And these tissues do not fix well because the original structure has been altered by changes brought about by lysosomal degradation. Now in this lecture, I will also introduce you to newer concept of programmed cell death because there have been a lot of changes in this area in the last few years. Last time we saw how there is hydropic degeneration or cloudy swelling because of cell injury. That means because the cell is injured, there is no sodium potassium ATPase, and so the cell swells up. Swollen cells can affect function, as you can see in this normal liver. If you normally inject India ink, it comes to the portal tracts, flows into the central vein and goes out. It doesn't stick around in the sinusoids because there is open sinusoids. Whereas if you have cell swelling because of injury, if you inject India ink, the India ink tends to stick around in the sinusoids because the swollen cells have obstructed the sinusoids. So the two forms of cell death were originally divided maybe 20 or more years ago into oncosis, which is cell swelling, and apoptosis, which is cell shrinkage. So oncosis we have covered in detail and ends in necrosis. Apoptosis, you can see in this diagram that it involves cell shrinkage, breaking up of the cell into small blebs and particles, which can be phagocytosed. So why do we have this kind of endogenous death programs? When organisms evolved multicellularity, it was a very long time ago, maybe 700 million years ago. There was a need to have control over the number of cells. That means that you need homeostasis of the cell number. You have to balance new cells produced and damaged and unrequired. So you can imagine that in a bacteria, all the organisms are competing with each other. They all want the nutrients and neither of them is willing to let it go. Whereas in multicellularity, you need to have control over both cell division and cell death. Cell division, I'm sure you have studied in detail in your physiology and biochemistry. Cell death is typically taught in pathology, although you might have been introduced to many of these concepts earlier. So if you look at the regulatory genes, some of these sense altered cellular environment and external stimuli, which in turn stimulate mitosis, like for example, release of growth factors. Others can inhibit mitosis. So rather than have mitosis, you inhibit it so that the cells do not multiply. But other forms of stimuli initiate apoptosis. or alternatively other forms of regulated cell death like pyroptosis or autophagy. Why do we have this fight between living cells and dead cells? You can easily imagine. There is no altruism, but altruism has to come in to maintain a homeostasis. Uh, Maybe 2 billion years ago, eukaryotes evolved at the end of one glacial period because cyanobacteria converted all the carbon dioxide in the atmosphere to oxygen, and it became necessary to evolve organisms which are efficient at using oxygen. And therefore, certain microorganisms evolved what we now call as the mitochondrial membrane transport. These were originally free living organisms which were taken up by other prokaryotes and they become eukaryotes. So, eukaryote is an endosymbiosis between a mitochondrion, which was originally free living, and other prokaryotes. Eukaryotes can be unicellular, and for almost a billion years they remained unicellular. Some of them, of course, got plastids and became capable of doing the work of both the cyanobacteria and the mitochondria to evolve into something more, that is a multicellular organism. There was a kind of a fight. You can say that the two genomes, that of the mitochondria and that of the protozoa, the protozoan genes kind of lost out. It ended up that cell division in cells end with the mitochondria being retained, but the genome of the uh, host cell getting exchanged. So the mitochondrion itself is conserved, but it ensures that its host exchanges genome so that the host evolves better. Similarly, when it came to multicellularity, again the same thing happened in meiosis and in sexual reproduction. The host ensured genetic exchange, but the mitochondria in the egg, they prevail. So the mitochondria remain the same, but new genetic material comes in. When it came to cell death, also there was a struggle between these two, and it turned out that the mitochondrion remained at the site of all the events related to cell death. So the further back you go, more you find that the endogenous death or programmed cell death programs are integrated initially from the mitochondria. Later on, more complicated and more extensive forms of regulated cell death did evolve, but initially they were mitochondria centric. Today, of course, we have come a long way, 700 million years of evolution. And one of our biggest problems is uncontrolled cellular proliferation causing cancer. So, multicellularity with unregulated mitosis or unregulated apoptosis causes cancer. We also have excessive levels of cell death. There are a large number of diseases which you will subsequently be covering, like Alzheimer, Parkinson's, rheumatoid arthritis, etc. So why are we learning this? Because there are a lot of conditions which are associated with apoptosis, which are listed here. The loss of tail in humans, some of the chemoresistance genes, cancer development, targeted therapies, all of these are involving apoptosis. If you look at an overview of apoptosis, what it means is falling off leaves. And it was first discovered only in 1972, which is pretty late because cell injury was being described by Rudolf Virchow almost 100 years before this. It is a programmed cell death which is isolated, imperceptible, and follows a standard, tightly regulated process. There is activation of cytoplasmic and nuclear enzymes which cause cell shrinkage and break up into apoptotic bodies. It's a very fast process. It happens in cell culture within minutes. In human tissue also, it can be in minutes or maybe 20 minutes and is very quickly ingested by macrophages which are attracted there. There are physiological and pathological reasons for apoptosis, and apoptosis can be triggered by both intrinsic or extrinsic reasons, but they all follow the same common pathway. Rather than tell you the step by step evolution of apoptosis, I will go backwards. I will tell you what it looks like first. So, you saw this necrotic cell last time, where the cell membrane has disappeared and has been lysed because profound cell membrane disturbance is at the core of cell injury. Nucleus is just irregularly fragmented, cytoplasm is all digested by lysosomal enzymes. Look at the difference with an apoptotic cell. The nuclear membrane has got all the chromatin condensed on it. The cytoplasm has shrunken and is regularly organized into cross-linked proteins. You can see in this picture from Robbins how the nuclei can have this crescentic appearance of nuclear chromatin condensed towards the center. You can see the cytoplasmic blebs, nuclear debris. Here the nucleus is fragmented to multiple debris like this. If you look at tissue, you find that there is condensation of the cytoplasm and presence of nuclear pycnosis. So the pycnosis part looks similar. The cytoplasm becomes pink and shows presence of nuclei which are fragmented. See how the cell has detached from its neighboring cells and has rounded up. If you induce apoptosis in mouse myocardium, you can see what a large cell it initially was. This is a myocardial cell. And now this area, which originally had a myocardial cell, instead is occupied by this large number of macrophages which have come to ingest it. And the nuclei of all the fibers which were here have turned into these tiny little apoptotic bodies which are visible here. If you look at this is normal thymus full of lymphocytes which look like this on electron microscopy. Whereas here you have given dexamethasone which induces apoptosis of the lymphocyte. So all the small condensed nuclei of the lymphocyte are the morphological counterpart of apoptosis. If you look at the EM of the same, you find that these nuclei, which were originally round, have assumed other kinds of shapes. Some of them have started fragmenting. The nuclear chromatin is more condensed. Here it is much more condensed and shrunken like pycnosis. Here there is a large cytoplasmic bleb, which is a fragment which is detached and is lying free. Here also you can see these are the fragments of the lymphocyte which have undergone apoptosis. Sometimes you can see that these cytoplasmic fragments contain organelles and their debris within them. And there seems to be an organized, it doesn't look moth-eaten, it looks as if it is like a organized structure because of the cytoskeleton aggregation. Here also you can see the nucleus is condensed. Here a macrophage has come. A macrophage has ingested all these nuclei, 1, 2, 3, 4 nuclei and some cytoplasmic debris. Here you can see that these are the apoptotic lymphocytes. This is also having nuclear condensation. Here these are extremely condensed and pycnotic nuclei. One of the features of cell injury was ribosomal disaggregation, whereas in apoptosis you can sometimes see, rarely, ribosomal aggregations. Here is an apoptotic renal tubule. Compare with the necrotic tubule which I showed earlier in the last lecture. In that the entire outline was preserved but all the nuclei were lost or all the nuclei look pycnotic. Here most of the nuclei are preserved and then suddenly you find an apoptotic body over here. And you can find occasional nuclear dots here. These are possibly represent other cells which have also undergone apoptosis. Other tubules which are nearby look relatively normal, but this one also has an apoptotic body in it. So possibly they gave some kind of an apoptotic signal like tying the artery or something like that. And this is the corresponding EM. On EM, you can see this crescent shaped nuclear condensation just under the membrane. An apoptotic cell like this in an epithelium, say of the enterocyte, once it has gone on apoptosis, it shrinks. And two things can happen. Either a macrophage can come, here it is squeezing through pores in the basement membrane, a process called apoptosis, and it is coming in and it is eating it. After that, it will squeeze out and take it away. Or alternatively, there can be lysis of these cell junctions and the apoptotic bodies which will undergo fragmentation can be extruded out. To summarize the morphology of apoptosis, there is cell shrinkage, detachment from cell junctions. They turn pink, and in pathology, as you study many topics, like for example in liver, you will call it councilman bodies. Similarly, lots of names are given to the bodies which you get in different types of diseases. They have a dense eosinophilic cytoplasm. There is early condensation of chromatin at the nuclear membrane, followed by nuclear fragmentation and further condensation into small, tiny pycnotic blobs. Cytoplasmic bodies containing the organelles bud off. There is extrusion of the membrane bound cytoplasmic bodies. So because they are membrane bound, the proteins which are within these cytoplasmic bodies do not expose the damps and therefore do not elicit an inflammation. Additionally, there is expression of cell membrane recognition molecules which attract macrophages, and in lymph node you call them tingible body macrophages, because in lymph node apoptosis is very common. All the self-reacting or not very having much affinity kind of lymphocytes are removed by apoptosis, which are immediately phagocytosed by these tingible body macrophages, which reside in the lymph node all the time, and they use these recognition receptors. So this is a summary of the differences between apoptosis and necrosis. Apoptosis involves single cells or small clusters of cells. It is different from necrosis, which is often contiguous. Apoptosis has shrinkage and convolution, whereas necrosis has cell swelling. Pycnosis and karyorrhexis can be there in apoptosis, whereas necrosis shows karyolysis, pycnosis, and karyorrhexis. Apoptosis has an intact cell membrane, and so the cytoplasm is retained, whereas necrosis has disrupted cell membrane and the cytoplasm is released. There is no inflammation in apoptosis, whereas in necroptosis, inflammation is usually present. The apoptotic process was first elucidated in this organism, the C. elegans, which is a nematode worm, which has precisely 1090 somatic cells. Exactly 131 of these at different points in development undergo programmed cell death. And if you study it from worm to worm, it is completely invariant. With remarkable accuracy and control, the same cells at the same time point will undergo apoptosis. And therefore, apoptosis was elucidated by looking at the control proteins which were involved in C. elegans, and subsequently homologs when humans were found. Now, of course, we have sequenced the whole genome and we know the control in great detail. But look at this. Unlike 131 cells in this worm, in the adult human body, 10 to the power 9 cells, that's about 10 billion cells, undergo apoptosis every day. Every day, these many cells are produced. Every day, these many cells are removed. Now, one way to study apoptosis is to go from what starts it, but I prefer to go at what is the end point. Apoptosis always has the same execution phase. Apoptosis is executed by caspases, which are termed as executor caspases, which are 3, 6, and 7. Their function is to cleave the cytoplasmic and nuclear matrix proteins and the cytoskeleton. And the executor caspase also activates this transglutaminase, which cross-links all the proteins in the cytoskeleton. There can be activation of endonuclease or DNAs and cross-link them. The cross-linking is carried out by transglutaminase, which cross-links all the proteins in this cytoskeletal. Executor caspases also cause activation of endonuclease. These are called caspase 3 activated DNAs, and they work similar to topoisomerase to produce dual strand cleavage. But unlike many other topoisomerases, they cause an internucleosomal cleavage. And so because it is internucleosomal, there is every nucleosome has the DNA wound around it, and around 180 base pairs are wound around each nucleosome. And by cleaving in between the nucleosome, you get a ladder pattern of electrophoresis. That means all the DNA fragments are multiples of 180. Another function of the executor caspase is phagocyte recognition. So phosphatidylserine and thrombospondin are expressed on the cell membrane. If you do an electrophoresis, if you have a necrotic pattern, the DNA is cleaved into all possible fragments, because the DNA which is activated is an endonuclease which cleaves all fragments. Multiple types of lysosomal DNases and endonucleases are activated, whereas the caspase activated DNase cleaves in a ladder pattern. So all the fragments have multiples which give rise to this ladder pattern, and this is the human genome without cleavage. So all of them are large DNA which do not migrate on electrophoresis. The second step to understanding of the apoptotic pathway is to look at what causes activation of execution caspase 3. There are two ways it can be activated, which is caspase 8 and caspase 9. And these two have different pathways for activation. Extrinsic triggers cause final step caspase 8 activation. So this can be initiated by fast ligand binding to fast receptor and fast associated dead domain activation. It can be TNF alpha binding to TNF alpha receptor and the TNF alpha receptor associated dead domain activation. It can be by perforin granzyme. Many other triggers evaluated in cell cultures and in living animals, one of which is ceramide, but there are many others, also can activate the extrinsic triggers. And the extrinsic pathway also has some inhibitors like the flip protein. So some viruses and cells can express more of this flip protein or its analogs, which prevent apoptosis. The other pathway is intrinsic. So intrinsic, there is a mitochondrial trigger, the final step of which is activation of the other enzyme, which is caspase 9. This involves activation of APAF1, which forms an apoptosome. We'll see the details of this. And APAF1 is in turn activated by release of cytochrome C from the mitochondria. So the intrinsic mechanism is related to mitochondrial trigger. There is an integration phase. The integration phase is integration of pro-apoptotic and anti-apoptotic signals. Pro-apoptotic signals from the nucleus include proteins like BAX and BAC or BAD. Anti-apoptotic signals include BCL2 and BCLXL, and these in turn may be related to the expression of activity of growth hormones binding to their receptors, expression of P53, growth arrest and DNA damage or GAT45, etc. We will see further details of this in the subsequent slides. All of these BACs, BAC, BCL2 typically form oligomers or dimers, and these dimers affect the mitochondrial permeability. So BCL2 stabilizes the mitochondrial membrane by forming BCL2 dimers, whereas if a BAX BCL2 dimer is formed, it doesn't stabilize the mitochondrial membrane, opening the pores and causing release of cytochrome C, which, as you know, is a part of the electron transporter. The APAF1, on the other hand, forms this large complex called the apoptosome. This apoptosome is a complex method where multiple proteins assemble. there is cascade activated domain or cards which are generated, and these in turn have both inhibitory and pro-apoptotic signals integrated on the apoptosome. So I made this simplified diagram. I know it looks complex, but it's actually simplified compared to many of the others which you might find. This big box on the outside is the cell. This round over here is the nucleus. This round over here is the mitochondria. Now, the end result is executor caspase activation, whose results we have already seen. So protein cleavage, cross-linkage, phosphatidyl serine expression, and internucleosomal cleavage of DNA. We'll go into some more details subsequently. What activates executor caspase is initiator caspase 8 or 9. 8 is because of extrinsic signals. So an extrinsic signal could be it could be fast ligand, or it could be a cytotoxic T cell or a natural killer T cell signal. So when a cytotoxic T cell binds to cell recognition receptors, it releases perforin, which punches holes through the membrane, and the granzyme B gets internalized into the cell. Granzyme B can directly activate initiator caspase 8 and also directly activate executor caspases and other caspases like 7 and 10. So granzyme B has very complex activity, a little different from the others which are shown here. On the other hand, this is a nuclear integration. So in the nucleus, you can have BCL2, which prevents cytochrome C release in the mitochondria by stabilizing the membrane, or BAX-like proteins, which promotes the cytochrome C release. If there is an injurious agent, like we saw in the previous lecture, and there is DNA damage, P53 is expressed, or GAT45 also, which via P21 inhibits the CDKs or the cyclin-dependent kinases, and there is a cell cycle arrest. The arrested cell tries to repair. If the DNA is repaired, MDM2 is released, which inactivates the P53. DNA is not repaired, BAX is released, which causes apoptosis. This is prevented by the balance between BCL2 and these BAX gene. Similarly, growth factor receptors can work in two ways. Once it binds to the receptor, signal transduction by various methods takes place, which via transactivating factors can either express BCL2, or in the absence of signal, there could be a BAX expression. And if there is BAX, which will promote mitochondrial release of cytochrome C. Once this is released, the apoptosis is formed, which causes cleavage and assembly of the initiator caspase 9, which activates the final cascade. So apoptosis is not as difficult as you might think until you come to this slide. You can see the number of things which are involved in apoptosis. We have alpha 14 activation by caspase 3, which cleaves the cytoskeletal proteins and cross-links, causing loss of overall cell shape. Gelsolin is activated, which causes depolymerization of actin, and therefore there is plasma membrane blebbing. It inactivates the retinoblastoma gene, which is a part of cell cycle regulation, and so it gets arrested or dysregulated. So inhibitor of caspase activated DNase is removed by the caspase 3, and therefore you have internucleosomal cleavage. Internucleosomal cleavage is caused by the caspase activated DNase. ATP11A and ATP11C, which is also called as the phospholipid flippase. The reason it's called phospholipid flippase is because it irreversibly flips the phosphatidylserine, which is normally expressed on the inner mitochondrial membrane, to the outside, which exposes it for phagocyte recognition. Similarly, XKREL-P8 is activated, which is also called as phospholipid scramblase, which also causes phosphatidylserine exposure. Transglutaminase are activated, which cross-links the cytoskeleton, and this polymerization prevents leakage of the cellular contents. It can inactivate PARP or poly-ADP ADP ribose polymerase, which is an ATP-dependent DNA repair mechanism. So this is an important function. It makes it possible for the cell to conserve ATP for apoptosis. Apoptosis is an ATP-dependent process. It requires a lot of ATP for all these enzymes to function. If a cell is ATP deficient, you cannot have apoptosis. It can inactivate focal adhesion kinases, which cause cellular adhesion, and therefore there is detachment of apoptotic cells from the ECM and neighbor cells. Lamin A can be cleaved. So these are nuclear envelope scaffold proteins, and cleavage causes nuclear shrinkage and fragmentation. Therefore, there is removal by phagocytes. So apoptotic bodies are rendered bite-sized. There are changes in membranes which exposes receptors like phosphatidylserine of the inner leaflet of plasma flips out. Others like annexin I, calreticulin, thrombospondin are expressed, which serve as eat me signals, and the dead cell disappear within minutes without a trace. That's why we never found them for a long time. And therefore, there is no secondary necrosis or inflammation. There are ways to mark apoptotic cell. One of them is tunnel, which is TDT or nick end labeling. So nick end labeling means that wherever the DNA is nicked, you can label the end using the TDT or terminal deoxynucleotidyl transferase, which is used with a biotin labeled UTP. And you can detect the biotin with a color reaction. So this is, you might have doubt, this is a seminiferous tubule, is this a normal cell involved in spermatogenesis, or is this apoptosis? If you mark it with tunnel, you know that it's apoptosis. Similarly, in this cardiac muscle, they can stain the apoptotic debris with tunnel. Or you can directly label it using caspase 3. So caspase 3, when it is activated, can be specifically labeled like this. So there's a large myocyte here in which there's a lot of caspase 3 activated, which is staining. These are not active. And here you can also see that the apoptotic bodies, so much smaller than the large myocyte, can also be labeled. Another is anti-phospho H2AX, which is a histone variant which becomes phosphorylated during apoptosis, or all of the other membrane cytoplasmic enzymes which we discussed earlier. They can also be used for labeling it. You can do a real-time PCR analysis of all the BAX, BCL2, and other control genes which regulate apoptosis. An interesting way to detect apoptosis is by looking at the membrane alteration. So you can detect the phosphatidylserine using annexin V, which binds to it, and you can detect it by either fluorescence or flow cytometry. The advantage is it's very sensitive. Even a single cell amongst a large cell culture can be detected. You can see how they have labeled it. The disadvantage is that sometimes necrotic cells are labeled. Similarly, even tunnel, sometimes randomly cut necrotic cells are also labeled. So it's a good way to differentiate between a lymphocyte and an apoptotic cell. Not so good to differentiate necrotic cells. So the best way to recognize that is by caspase 3 labeling or activated caspase 3 labeling. Another way to make it out is looking for the membrane integrity. Necrotic cells don't have membrane integrity. So various kinds of dye exclusion tests can be complementarily added to the flow cytometry, like tropidium iodide, trypan blue, etc. There are complex mitochondrial assays available. So you can use laser scanning confocal microscopy to directly look at mitochondrial changes. Is it swelling? Is it leaking? Is it not? By using submicron optical slice of living cells. Another is to measure electrochemical gradient using lipophilic dyes, which either accumulate in the mitochondria if there is healthy mitochondria, or accumulates in the cytoplasm if the mitochondria are damaged. Let's now come to where all you get apoptosis. Some are physiological. So it is a part of normal homeostasis. having complementary but opposite role to mitosis. And as said before, 10 billion cells are made each day just to balance those which are dying out. It's an integral part of development and embryogenesis in the nervous system, immune system, formation of digits, interdigital spaces, regression of tail. There is remodeling of the adult in things like follicular atresia in the post-ovulatory follicle, sometimes in the mammary gland involution, in wound healing, evolution of granulation tissue into scar tissue, in involution of hormone dependent tissue like endometrium. When the progesterone simulation stops, the endometrium undergoes apoptosis. In breast, after lactation, it involutes using apoptosis. It's also important in getting rid of pathogen invaded cells and eliminated activated or autoaggressive immune cells. Pathological apoptosis, on the other hand, is used for elimination of damaged cells. For example, misfolded stress proteins can cause either cell injury or in wound healing, where excessive scarring and fibrosis can be there if apoptosis is not properly controlled. It is important in cytotoxic T cell and natural killer T cell mediated antiviral activity because of the perforin granzyme mechanism. In pathologic atrophy, for example, in atherosclerosis of blood vessels supplying the kidney, in hormonal atrophy of prostate, which might be because of excess hormones getting reduced or because you do chemical castration. If the duct is obstructed, for example, the exocrine pancreatic duct is obstructed, the pancreas undergoes apoptotic atrophy. It's also a significant component of major diseases. It might be because of too little apoptosis, like in cancer or many autoimmune diseases where the self-reacting lymphocytes are not eliminated, or there could be excessive apoptosis, like in AIDS, ischemia, and neurodegenerative diseases. In apoptosis, it's a central mechanism in cancer. Reduced apoptosis is seen by increased BCL2, typically, like for example, certain kinds of lymphomas. It is also involved in evasion of immunosurveillance. So the T cells and natural killer cells, they try to destroy the tumor cells, but the tumor cells can evade by either reducing the expression of fast receptor or upregulating something called PDL1, a programmed death ligand 1, to induce energy in the cells which attach to them. You'll study these in detail later. Another thing which happens is mutation in P53. P53 activates DNA repair and holds the cell in cell arrest. So bypassing this is critical for cancers, which therefore blocks the apoptosis. Similarly, upregulation of PI3-AKT pathway, which regulates apoptosis, makes it possible for these cells to survive despite the presence of cell death signals. Apoptotic mimicry is very interesting. Parasites and viruses expose phosphatidylserine on their surfaces, and therefore they can hide by masquerading as apoptotic debris. So this could be classic apoptotic mimicry in which actual phosphatidylserine is expressed, like for example, dengue virus, vaccinia, Ebola. They hijack the endocytotic mechanisms for internalization and can express the phosphatidylserine on the membrane which they possesses, which they derive from the host cell. Non-classical, you need mimicry of the phosphatidyl itself because there is no membrane, and therefore you can have structurally similar viral proteins. There are a number of methods to inhibit apoptosis. One of them is flip and BCL2, and there are inhibitors of apoptosis proteins or IAPs, which typically inhibit the apoptosome. We saw that the human apoptosome has this complex structure. Similar complex structures are seen in fruit flies and in nematode, that is C. elegans. And as we started understanding cell death further and further, we initially having thought that plants and yeast cells do not have apoptosis, that they had evolved other methods of multicellularity, realized that there is some amount of programmed cell death, especially this mitochondrial permeability transition pore. So this is at the center of apoptosis and other kinds of cells. Okay, and all kinds of stresses can induce plant cells also to undergo programmed cell death, which could be related to cytochrome C release and calcium influx. The difference is that the classic mechanism, that is the caspases, are not there. So the cells shrink but without breakup. The caspases are not there. But despite that, there is a ladder-like pattern on electrophoresis because of the endonuclease activation, which does take place. So you can divide it into apoptosis-like, which is very fast but slower than mammalian cell apoptosis. There is an autophagy-like, very slow cell death, and a necrosis-like cell death, which is slower cell death in cultures after you give injury to the plant cells. Having understood that there is a conserved and complex mechanism of cell injury, now people look at cell death as accidental or programmed or regulated. Accidental cell death is what you studied in cell injury, which is manifested as necrosis. We studied it in detail. Regulated cell death is similar to apoptosis, but there are other types like necroptosis, pyroptosis, etc. They occur in the absence of these exogenous environmental disturbances and are part of the built-in effector cells to control tissue proliferation and turnover. It is what is involved in normal elimination of useless or potentially dangerous cells, and they may or may not have inflammatory depending on whether they have a necrotic morphology or a apoptosis like morphology. So if you look at regulated cell death as a class of cell death, you can have an apoptotic morphology, which is like the apoptosis which I just explained to you, or it can have a necrotic morphology. And there are, just like there are 12 o'clocks, there are 12 types of apoptosis. There are even more, but this is the one which is listed in a recent review. Let me go into some of these. First of this is pyroptosis. Pyros means fire, and some people thought that the cells tended to flame out rather than shrink. That's how it was first described. It is also called pyros because of fever, so the temperature rises. This can be thought of as a programmed cell death with collateral damage and inflammation. It was first described in macrophages infected with salmonella. It is also caspase dependent, just like apoptosis, but there is also release of pro-inflammatory cytokines. There is presence of nod-like receptors and toll-like receptors, which are pattern recognition receptors, which recognize this salmonella or other PAMs or pathogen-associated molecular patterns in microbes, and a card domain is there which activates an inflammasome. The inflammasome, like the apoptosome, is a complex assembled protein structure, also called the NLRP3, which cleaves procaspase 1. Once caspase is formed, caspase forms pores in the plasma membrane by this molecule called gasdermin. It also causes release of interleukin 1 beta and IL-18, which attracts the neutrophils and causes fever. The pores cause cell swelling. Just like you saw in necrosis how there is cell swelling, these pores also cause cell lysis, which releases the dams, and therefore there is even more inflammation than what the interleukins attract. Nuclear integrity, however, is maintained because there is no internucleosomal fragmentation. It is not caspase 3 mediated. There is no nuclear fragmentation. So it's a different morphology. The second important one is necroptosis, which is programmed necrosis, you can think. This is because of an activation of necrosome. The necrosome is an alternative to the inflammasome, which has two components, the RIPK3 and the MLKL. So the RIPK3 is also called the ripoptosome. It is like a rheostat for apoptosis versus necroptosis. So if the ripoptosome finds that the caspase 8 is inhibited, then it will cause necroptosis. So there are multiple triggers for necroptosis. Some include reactive oxygen species, DNA damaging agents. All the things which cause cell injury can also activate necroptosis. Interestingly, it is seen in CMV infections because the CMV has a caspase inhibitor. So it inhibits the caspase 8, and therefore human body uses an alternative method to eliminate CMV infected cells. In all of these, the trigger is via the death receptors like FAST, TNF receptor, TRAIL. It can also be triggered by toll-like receptors or pattern recognition receptors. People have therefore come up with oncosis as a switching mechanism between necrosis and apoptosis. And the two forms which we have studied, necrosis and apoptosis, are two opposites of an extreme opposites. And there are possibilities that there could be programmed cell death with in between processes. One type of cell death was initially called Anoikis because at that time people did not realize what were the underlying mechanisms. Anoikis means loss of home or homelessness. Whenever cells detach from the extracellular matrix or the basement membrane, they underwent apoptosis. But at that time, because they didn't know it was apoptosis, they called it Anoikis. So it can use both the extrinsic and the intrinsic apoptotic pathways, and there is initiation by the laws of endocrine dependent anchorage. It's important in your topic of um, malignancies and neoplasia that you always have cell survival signals. Beta catenin, for example, is an intracellular as well as an extracellular molecule, and it protects against anoikis. If there is detachment, beta catenin signals promote apoptosis. NF kappa B activation constitutively prevents apoptosis. It's a cell survival signal. Same with the PI3 and MAPK3 pathway, which all either cause degradation of pro-apoptotic signals like BIM or upregulation of the anti-apoptotic BCL2, etc. Escape from anoic is essential to carcinogenesis. So you get anoikis resistant malignant cells, which might be because there is flip overexpression or overexpression of EGFR, or there is a ligand independent EGFR which is constitutionally active. So cell survival signals are constantly delivered. There could be hyperactive growth signals, and there is also this EMT, that is epithelial mesenchymal transition, which means that normal epithelial cells which are polarized become more like mesenchymal cells. There is migratory capacity, invasiveness, and even if it is detached from the ECM, it is resistant to apoptosis. And this is controlled by something called as a cadherin switch. So instead of the E or epithelial cadherin, there is N cadherin expression, which has a that is a mesenchymal protein. Autophagy is a process you will be taught in detail in one of your subsequent lectures. There is always a question, is there autophagic cell death? Now, autophagy is because of certain genes called ATGs or autophagy related genes, and it is expressed whenever there is cellular stress to recycle the cytoplasmic components and degradation of organelles. So, whenever there is stress in the cell, these ATGs are going to be expressed and autophagic process are going to be there. Is it just a bystander? Is it just associated with autophagy? Or is it mediated by autophagy? In mammalian cells, we are not sure. In plant cells, autophagic cell death is an important type of cell death, and so it is in certain other situations, but not probably in mammals. There is a cell death called entotic cell death. This is a non-phagocytic cell cannibalism. Neighboring cells eat the other neighboring cell. It requires autophagy pathway proteins, and there is no caspase 3 activation. Instead, it is mediated by release of lysosome or phagolysosome formation. So one cell swallows another and digests it. That is entotic cell death. Another type of cell death which you will cover in detail in inflammation chapter is netotic cell death. Net is neutrophil extracellular traps, and this is because the nuclear material of the neutrophil is extruded out to trap the pathogens and help in their killing. The killing is also done by oxygen species released by the neutrophil. So there are two types of netosis. One is suicidal netosis, where the neutrophil lyses One is vital netosis. The neutrophil slowly moves nuclear material out via vesicles until all the nuclear material is gone and then the cell itself dies. So that is called netotic cell death. There is one type of newly described cell death called ferroptosis. Now, in this, there is a iron non-enzymatic lipid oxidation by Fenton reaction. We already saw Fenton reaction in our previous chapter. So lysosomal Fenton reactions can cause a formation of membrane pores by severe lipid peroxidation. So you can remember that in one word, that this is pyroptosis is lipid peroxidation with pore formation initiated by free radicals generated by Fenton reactions. Certain proteins protect against it, and you can also inhibit it by using iron chelation and iron overload. So for example, in disease like hemosiderosis, these kind of cell death take place. Another unusual kind of cell death is parthenotos. This is important in stroke, neurodegenerative disease, and spinal cord injury. Whenever there is ATP depletion, you cannot undergo energy dependent apoptosis. So these are alternate method of cell death. It is mediated by PARP or poly-ADP ribose polymerase, a nuclear enzyme. It senses mild nuclear damage and recruits base excision repair mechanism by PAR polymer formation. In PARP mediated cell death or parthenotos, there is hyperactivation of PARP1 due to profound DNA damage. The PAR polymer accumulates, translocate the AIF protein from mitochondria to nucleus, and there is large scale DNA fragmentation, and that's how the cell dies. There's another form, yeah, it doesn't end. Lysosome-dependent cell death, which is because of release of lysosomal membranes by permeabilization. So the lysosomal contents are released in the cytoplasm, and these lysosomal enzymes belong to the cathepsin family. So there is lysosomal dysfunction or lysosomal membrane permeability. In lysosome, normally the cathepsins are inside. If the cathepsins come out, it could be either partial, where there is selective release of cathepsins which attack mitochondria, or total lysosome rupture which causes massive release of enzymes and uncontrolled cell death. Immunogenic cell death is simply cell death which generates an immune response. Remember that in apoptosis there is no generation of immune response. So here this is a regulated cell death where there is a activation of adaptive immune response, and this is specifically a kind of an induced cell death. So in viral infections, when you give chemotherapy for cancer or phototherapy or radiation therapy, etc., you are trying to generate immune responses because they have anti-cancer effects even against the living cells. And the purpose of this is to expose immunogenic signals like ATP, HMGB1, heat shock proteins, which are all DAMPs. And you do that by delivering ER stress. So this is a kind of cell death where there is excessive ER stress. It could be physiological, but by and large, we use it for these kind of chemotherapeutics. MPT or mitochondrial permeability transition driven necrosis is what I discussed in plant cells, where there is intracellular oxidative stress followed by cytoplasmic calcium overload, and it causes a necrotic morphotype. These are some of the references of the what I explained. So you might think that I've gone into too much detail of cell death. Actually, that's not the case. Okay, you might be feeling like these cells, because cell death is not very pleasant. There are too many different types which are confusing. But what I've tried to do is make it into small bite-sized fragments which you can swallow. So like apoptotic bodies, they are easy to phagocytose. To summarize, we discussed apoptosis in detail about extrinsic apoptosis and intrinsic apoptosis. These are two which you need to know well. Apart from these, you should know a little bit more about necrosome and necroptosis and pyroptosis. So you can remember that the pyroptosis is because of the NLRP or the nod-like receptors, the toll-like receptors, formation of inflammasome, activation of caspase 1 and interleukin 1 release. Whereas the necroptosis is because of formation of a necrosome mediated by the RIPK3 and MLKL. And this happens when there is caspase 8 inhibition. Apoptosis, or especially extrinsic apoptosis, because of caspase 8 activation. Necrosome is because of caspase 8 inhibition and an alternate pathway of cell death. So the different types of cell death can be grouped into apoptotic morphology, or regulated cell death can be grouped as apoptotic morphology and necrotic morphology, and the two have various forms in between. Thank you very much, and I'd like to acknowledge the postgraduate seminars of Christabella Thomas and Hina Kandakar, who presented this earlier in the year. Thank you very much.